All right, so remember, I'm going to do this 3 times tonight. Good evening. Canada is an Arctic nation, yet only a small percentage of Canadians actually live there. How do people living in the southern parts of Canada, or anywhere else in the world for that matter, learn about the unique and fascinating marine animals that inhabit this enigmatic environment? Let me present to you the online Arctic Marine Life course. The course is supported by the Science in Canada's North Café Scientifique series, and we would like to acknowledge the support of the W. Garfield Weston Foundation and the Canadian Association of Science Centres for their support. Our partners include the Alliance of Natural History Museums of Canada, Canada's accredited zoos and aquariums, TELUS Spark in Calgary, the Canadian Museum of Nature in Ottawa, and the Discovery Center in Halifax. Keep track of the dates and locations of all of our presentations on our website, www.vanaqua.org/Arctic Marine Life course. If you miss a presentation, you can always watch it online after it's been recorded. If you have any questions about the course, or you would like to register, you can contact me at jonathan.holtquist@vanaqua.org. Our speaker is Dr. Peter Ross, Director of the Ocean Pollution Research Program at the Vancouver Aquarium Marine Science Centre. Peter's research focuses on evaluating the effects of pollutants on the health of marine animals. He has collaborated on a number of studies conducted in the Arctic. His presentation will focus on benthic ecology and how pollutants can be used to map food webs. Dr. Peter Ross.
Thanks, Jonathan. Thank you and welcome everyone to this evening's lecture. I'm delighted to be here. I am not a benthic ecologist, but I can tell you that benthic ecology is the foundation for much of what I do when I look at ocean pollutants. So what I'm going to do tonight is explore the sediments and the sea bottom and provide a little bit of a segue into the invertebrates and into the fishes and the birds and the mammals that rely on the entire food web. And to do this, I'm going to take you 8,000 kilometers to the east of Vancouver and a little bit north up into Arctic Labrador. That will be the focus of this talk tonight. I'd like to acknowledge my co-author, Tanya Brown, who spent much time in this location, much more time than I did. And let me start by saying this is a very special place. It's a spectacular place that very few Canadians have visited. This is northern Labrador. If we look at that little yellow shaded region in this slide on the left, you'll see the Torngat Mountains National Park. This is a gem of a spot at the northern tip of Labrador, and it's been used for millennia by Inuit people who have relied on that interface of land and sea. You'll also note that this is not only an Arctic region. This is at the confluence of the Northwest Atlantic Ocean and the Arctic waters of Canada and the archipelago that we know of as the Canadian Arctic. So this area just to the east of here was also the site where the Franklin expedition traversed in the late 1840s as they sought the Northwest Passage and disappeared until we discovered a little bit more about what went wrong with their journey and their expedition. So spectacular spot, a spot that few get to, and a few people get to this part of the world because it's really tough getting there. It'll take you 2 days by air travel, or if you're doing field work there, you'll be fortunate if you can get aboard one of the Canadian Coast Guard icebreakers that transit these waters, coastal waters heading into the eastern Canadian Arctic. Or more appropriately, when you're dealing with nearshore environments and some of the coastal waters in these areas, working with some of the Inuit fishermen that know the waters very, very well, know the elements and know the creatures that move about these waters. And when we look at this topography, we as humans, we look at the land. That's what we understand. We are air-breathing animals. We stand on a shoreline. We look out to sea. We see the surface of that sea. And what lies below the surface of those waters represents a bit of a mystery to most of us. And it's that interface that I'd like to talk to you about today. We're going to go into the waters of a specific fjord in Labrador and try to understand a little bit more about why science, or why Canadians might be interested in this part of the world and interested in benthic ecology, for example. This is a creature I'm not going to talk about tonight, but I thought I'd put this animal up there because what Arctic talk would be complete without a polar bear? Of course, these creatures are marine mammals. They do rely very heavily on marine mammals and fish for their subsistence, and they can swim around these coastal waters with impunity. So not the focus of this talk, but certainly a charismatic creature that does tell us a lot about the health of our oceans, climate change, global pollutants, and the food web, which I'll be talking about a little bit more tonight. And when we go down into the waters, we immerse ourselves subsurface, and we go all the way down to the bottom of the sea. What do we run into? We run into substrate. We run into the sea bottom. And there are various ways of studying that. We can do that with remote technologies. We can study it by dropping in sediment grabs. We can, as you'll hear shortly, we can dive down with scuba gear, and we can have a look at that substrate. That substrate can be one of a number of different things. It could be soft, it could be hard, and it sort of, we're talking about something that is geologically and geographically defined. Ocean currents shape the way in which the substrate is formed and how it is characterized. And it's this substrate that forms the home for all the creatures that you and I would appreciate and understand, and upon which much of our fisheries resources are dependent. So this becomes habitat, geological, geographic, and ultimately into the biological communities that you're hearing about tonight. When we use seabed mapping, multi-beam sonar is one very powerful tool that is used extensively these days. That's simply being able to ping from a ship an array of sonar waves and getting a response back at the ship, which is then detected. And it's the time that it takes for that radar wave to hit the bottom and then bounce back up the ship that tells us how deep the water is, and it's the intensity of that signal that tells us something about the nature of the substrate, whether it's soft or whether it's hard. And we can start to create these wonderful maps that tell us about the substrate. And if we know about the substrate and we start to unravel or uncover the relationship between that substrate and the sea life upon which they reside and prosper, then we start understanding a little bit about home of those species. So in this picture here, the colorful one, the blue represents deep water basins that are often filled in with light sediments because of ocean currents and natural sedimentation. You see the lighter colors in the red, much shallower, more scoured, more exposed to ocean currents, probably harder and more rocky and cobbly than the deep water basins. And you'll see a little bit more about that tonight, but it's something you can follow up later. And of course, when we get this technological glimpse at the bottom of the ocean, we want to ground truth that. We want to make sure we understand what that technology is in fact telling us. And on the left is a sediment sampling device called a Smith-McIntyre. And that's something you drop on a line off the back of a vessel into the sediments. And it's like a shovel, and it will grab the sediments, and then you'll hoist them aboard the vessel, have a look at those sediments, and you'll be able to see very quickly whether it's soft, cobbly, hard, and you'll also get some samples of invertebrates in those samples. So using a combination of different tools, we can understand a lot about the sea bottom, the sea floor, which is habitat for a lot of different species. And in this case, if you send video down, you can have a look at the seafloor. On the left, you've got a soft bottom and you've got some brittle sea stars. On the right, you've got a hard bottom surface with all of the other species to be associated with those. And that's when you can start to combine the multi-beam technology with another backscatter signal coming out of the multi-beam and get these habitat-based maps. What species are likely to be found in these different types of sea bottom or seabeds. And if we look at sediments, let's get a little bit away from the hard bottom for a minute and just think about sediments. We can think about the creatures that live on the sediments versus the ones that live in the sediments. And we have both. There is a sediment food chain that thrives. And if you take samples of sediments, you'll see that. So we've got epifauna that will live on top of the sediments. You've got infauna that will be either inside the sediments or just at the sediment-water interface. So some of the bivalves and clams you'll find buried in the sediments. Others you'll find sitting on the top of the sediments or on a rock, filter feeding. And a lot of these creatures form the basis for the entire food chain upon which we will talk a little bit more in a second. So obviously some pretty spectacular creatures down there. I believe that you'll hear more about that in another course from this series, but some wonderful sea anemones, sea cucumbers, and other species, very colorful, very vivid. And if you think about these species and the way in which they interact with their environment, that's going to start to drive your ability to understand what kind of environment they might need. Do they need to attach to a hard surface like a rock? Just based on their lifestyle, or do they want to move around with their feet and their claws and their various means of locomotion? So understanding the way in which these species live, breathe, reproduce, move, and feed is going to dictate where they're going to find a happy life and a happy home. And that's that interaction between the seabed habitat type or substrate type and the creatures you're going to find in those locations. Of course, fish, we tend to understand a little bit more than invertebrates because we tend to have a lot more fish at the dinner table. We've got fish that rely on the bottom of the food chain and live on the seafloor. There are some flatfish. In this case, we've got the example in the upper right here, this shorthorn sculpin, which is a resident fish, lives on the bottom, eats a variety of different species of both fish and invertebrates. Pelagic species, of course. In this part of the world, we've got Arctic char, which is anadromous. It migrates from fresh water into the ocean and then back again, just like the salmon here. And char is, in fact, a salmonid. And this is something which is a little bit less connected to the sediments. But nonetheless, they're still looking for food. They're still feeding on some of the same invertebrates and or smaller fishes that we'll find more connected to the habitat associated with the bottom of the ocean. So even when we've got these mobile species that are only transiently present in some of these areas, they're still in some way connected to that seabed and connected to the sediments and the benthic biota upon which the food chain relies. I'm going to go up the food chain just a little bit here. Pigeon guillemots, this is an aquatic seabird, largely localized, pushed around a little bit by virtue of their desire essentially to find food, forage, and live in coastal waters in Arctic areas. But of course, when the ice comes along, then they're pushed out of some of their local habitats. They're still going to be largely a resident of the Arctic environment. So this is a chick. And for parts of this research, you can actually go out, and we've got colleagues at the Canadian Wildlife Service that go out and sample these animals, and then we can learn a lot more about their feeding ecology, their physiology, their nutritional status, and some of the anthropogenic stresses that we worry about when it comes to top of the food chain creatures, like climate change, food availability related to climate change, or global pollutants coming from multiple sources. Of Of course, the food chain would not be complete without putting a marine mammal at the top. I do like to talk about marine mammals. I've been studying marine mammals for almost 30 years. This is a ringed seal, and this species is very widely distributed throughout the Canadian Arctic. They're largely solitary. They don't hang out in big social groups. They're fairly small. They're long-lived, like most marine mammals, living maybe 35, 40, 45 years. And they really are very strongly associated with the ice edge. And you might say, wow, okay, marine mammals, they swim, you see them on rocks, you might see them on ice, you might see them on the ice edge. What's that got to do with the sediments? So as you can see with the story that I'm providing for you this evening, we're going from the bottom up to the top in terms of the abiotic environment, but also in terms of the biological environment. We're at the top of the food chain now. You might think that this creature is far removed from the bottom of the ocean, but in fact it is connected to the sediments in a number of different ways. And first and foremost, in terms of biological productivity. Think about the sediments being the foundation of what's going on in coastal waters. Think about all the creatures, the polychaetes, the sea urchins, the isopods, different invertebrates, different groundfish that are getting productivity associated with organic material in and around the sediments, and then think about that food chain. Then we're talking about a complete story, which in this case we're going to call the Labrador Coastal Food Web. And of course, a food web is a story about energy cycling, it's a story about carbon, it's a story about lipids, which is the currency of metabolism and growth, and it's also a story about pollution, and pollution is where I head next. In this case, Saglek Bay is a fjord in the middle of northern Labrador along the coast, just at the border of Torngat Mountain National Park, and there was an accident here. Well, one might say it was deliberate. There was a spill of 450 kilograms of PCBs in the late 1970s when the U.S. Air Force was transitioning to a Canadian military radar station, and a lot of waste polychlorinated biphenyls were bulldozed into the ocean as part of the cleanup effort. Unfortunately, out of sight is not out of mind, because this cleanup was not, in fact, a cleanup, and it led to a legacy of problems associated with its very persistent endocrine-disrupting chemical that most people have heard of. And despite being banned in 1976 in Canada, and in fact from much of the world, PCBs released in the 1950s, 60s, and 70s are still out there cycling around. So we're waiting for them to break down, which is very, very slow, but we're also waiting for them to get buried in the sediment with natural sedimentation processes. So that's the best we can hope for, is that PCBs really get sedimented out and buried, so there's a cap on top of these old reservoirs of PCBs. So a story here about the Cold War as well. This was a radar station during the Cold War to warn about possible incoming missiles or aircraft. And unfortunately, the legacy is related to this toxic chemical spill. And these are the sediment concentrations of PCBs. You see the red, that's the highest concentration of PCBs in the sediments at the bottom of the ocean. Why are PCBs found in the sediments? It's because they are not water soluble. And if they're not water soluble, that means they're fat soluble. And if they're fat soluble, it means they're binding to organic particles and silt and little bits and pieces of grains of organic material and dropping out into the sediments. Okay, so fat soluble, non-water soluble chemicals like PCBs, like dioxins, like DDT, a lot of the old persistent organic pollutants, or POPs, that you may well have heard about. These things are all fat-soluble, particle active. They bind, sediment out, that's why you find them there. And that's why we're worried about the food web. Because if they're particle active and dropping down into the sediments, the other place they can go is right into lipids, which means into algae, into bacterial cell walls, into the bottom of the food chain, and up the food chain. Because they're so persistent, they're very difficult to metabolize. So this map gives you an idea of PCBs as they spread throughout this localized fjord, Saglek Fjord. and contaminated vast stretches of this magnificent part of the eastern Canadian Arctic. And so what happens when we have high concentrations of sediment PCBs on the X or the horizontal axis? We see increasing concentrations in this fish, the short-fin sculpin. So a very distinct connection. This creature that lives at the bottom of the food chain and lives on the seafloor is not soaking them up through his or her skin. He or she is soaking these PCBs up from what it is eating largely. So we have a very direct relationship between the sediment contamination and the contamination of the fish, such that the areas that have the highest PCB concentrations have the most contaminated sculpins. Very simple story. Here's an interesting one. We've got guillemots. These are birds living on the surface of the water, nesting on cliff faces, swimming around, paddling around. Same thing. The birds that live close to the most contaminated sediment sites have the highest concentrations of PCBs. So another idea, another piece of evidence suggesting that contaminated sediments contaminate the food web, and these PCBs travel all the way up to these higher trophic level creatures. What does this mean for ringed seals? Well, ringed seals, much more mobile than the pigeon guillemots, so much more difficult to apportion blame, if you will. But in this case, we were able to show that those seals that inhabited local waters around the contaminated area, and this tall bar here represents the PCBs in adults and the PCBs in younger animals living locally versus those that went and fed offshore in cleaner, more remote parts of the northwest Atlantic Ocean. So much higher levels of PCBs in the seals that are feeding in areas around the contaminated radar station. And we worry about PCBs not because they're acute poisons, but because they're chronic, insidious, endocrine disrupting compounds, or EDCs. They are hormone mimics. Some of the PCBs, there are 209 of them, resemble thyroid hormone, and they can disrupt thyroid hormone physiology in seals, in rats, in humans. Okay, so endocrine disrupting compounds, that's a catch-all phrase or term for a lot of these persistent chemicals that have been largely banned, and those are the ones we call POPs, or the persistent organic pollutants. There are other newer endocrine disrupting compounds that are less persistent, but we are nonetheless concerned about the potential for chemicals to disrupt growth, development, and reproduction. You might have heard of bisphenol A. You might have heard of some other estrogen mimics. There are lots of them out there. But PCBs are these persistent chemicals that last for a long time out there. And what we see in these harbour seals, based on some very high-end molecular work, is with increasing PCB concentrations, we see different genes being turned on, essentially being upregulated here at about, well, at this dose, and it's about 1.5 milligrams per kilogram PCB. That's simply telling me there is a dose-response relationship. And a number of these, 1, 2, 3, 4, 5 genes out of the 15 or so that we looked at, were turned on by exposure to PCBs, which means that PCBs in this locally contaminated area are contaminating the food web, leading to high levels in ringed seals, and we're seeing molecular evidence of adverse injury or adverse effects. What this means to the health of the ringed seal or the population, more difficult to tell. This is molecular evidence, but what we do see in more contaminated areas are effects on reproduction, on incidence of tumors, on mass mortalities in marine mammals. We see a lot of evidence from much more contaminated marine mammal populations. Top of the food chain, contaminated ringed seals, and consequence for their health. So in very simple terms tonight, what I'm talking to you about is a food web or food chain. Food chain is a simplistic view of things, and that's what we've got right here. It's a story about who eats who, and this is a story about energetics and carbon and nutrients and the world around us, but it's also a story about pollution. And it's a story about some of these contaminants that do persist, that do bioaccumulate, and are toxic at the top of the food chain. Those are the three words you want to remember, PB and T, persistent, bioaccumulative, and toxic, because that's a generic definition of many hundreds, actually thousands of different chemicals. And that definition has a namesake in the form of the Stockholm Convention, which is an international treaty to phase out and ban the priority persistent organic pollutants. Very important legal framework by which countries buy in and promise to phase out, regulate, and take care of the environment by making sure they don't use these chemicals. Because these chemicals have no boundaries. They end up getting into these food chains and they present problems to biota. So if you're thinking about what I'm talking to you about this evening, you might have a few questions in your head. I'm touching on an awful lot of different disciplines, a lot of different topics. Each of these topics, each of these slides that I described this evening and spoke to this evening has a very rich compendium of literature, of works of knowledge that speak to such things as biogeography, physiology, toxicology, marine biology, a lot of different things going on here. So some of these questions can perhaps guide you in the right direction if you're looking to drill down just a little bit more to understand a little bit more about what I've spoken to you this evening. And some of those questions would be related to food webs. What is a food web? And what is the connection between the seabed and sea life on top of that? Why is there pollution in the Arctic? Why do we find pollution in the Arctic? Does it threaten wildlife or Inuit? Why are ring seals in particular vulnerable to high levels of these pollutants? And lots of questions here about tools. How do you study? How do you carry out research in the Arctic, where it's tough getting into it, into your study sites? The weather can be very challenging. The environment can be challenging. There's not much in the way of logistical support. You have to bring all your supplies with you safely, and you can't forget anything, otherwise you're going to be caught off guard. So what kinds of tools might be useful to apply to Arctic research? And in particular, if you're trying to understand something about mobile species, either anadromous fish or migratory birds or seals that may move along with the ice edge, how do you actually do that? And once you start thinking about some of those questions, there's a wonderful body of literature out there that can shed light on some of the interesting tools that have been applied with success to generate stories and insight about the Canadian Arctic. So thank you for your time, and I look forward to any questions you might have.
All right, thank you, Peter. So let's go ahead and take some questions from the audience. And Peter, maybe what you could do is just repeat the question so our online audience can hear it. So if anyone wants to go ahead.
So some of the genes that are being upregulated, what's like an example of one of the ones that have been turned on that maybe wasn't supposed to be?
So the question was, what are some of the examples of genes that were turned on by exposure to PCBs in this case? One of them is called the aryl hydrocarbon receptor, AHR. Aryl hydrocarbon receptor, we don't know a lot about it, but all mammals have the AHR. And in all mammals studied, what we see with the AHR is induction. So whether you're a lab rodent exposed to dioxin or whether you're a heavily exposed human, we will see the AHR turned on. So it's typically viewed as a very defensible biomarker of exposure to dioxin-like contaminants, including many of the PCBs. That was one of them. Another one was the estrogen receptor. You might have heard of estrogen before. So to have the estrogen receptor turned on by exposure to PCBs, which we've seen in killer whales here in British Columbia, which we've seen in harbour seals here in British Columbia, which we've seen in other species, is troubling, because that speaks to the potential for something like PCBs to affect the development of a healthy reproductive system in either males or females. So it's that sort of result that we tend to worry about. With humans, very difficult to study these things because we've got a lot of confounding factors, very complex diets, various backgrounds in terms of what we do and what our habits are. But those are two examples of genes that were turned on. Third one was thyroid hormone. Thyroid hormone receptor, and of course you know what thyroid hormones are, very important for metabolism and growth and development. That is another gene endpoint that has been routinely turned on by exposure to moderate to high levels of PCBs. Should point out though, in all of these cases the doses were relatively low. With these ring seals, they probably have a concentration of PCBs that on average would be 2 to 4 times higher than the average Canadian human. So relatively low by our standpoint, and certainly relatively low compared to a lot of other marine mammals in southern Canadian waters.
How common are PCBs outside of Arctic waters? Do we know that? Do we measure that, like in the effluent of the Fraser River?
So how common are PCBs outside of the Arctic? Very common. PCBs are everywhere. They were used extensively between 1929 and the mid to late 1970s. Because they're so persistent, they don't break down. They basically cycle around the world. They move through the atmosphere to remote areas. We find them, of course, throughout BC. Our resident and transient killer whales here in British Columbia are some of the most PCB-contaminated marine mammals in the world. And we're watching the concentrations of PCBs decline. Since regulations in the mid-1970s in Canada, we've seen a decline in PCB concentrations such that harbour seals probably have 1/5 the concentrations that in BC that they did 30 years ago. So we have been watching these declines, but PCBs do persist and continue, and the more we learn about them, the more we recognize that they were a threat that had to be addressed from a management and source control perspective.
We have a couple of online questions, but I need to get them. So if we could take one more question here, and then I'll get those 2 questions. Got another one? Great. Okay, awesome.
What were the PCBs being used for at the radar station? What contained them?
So what were the PCBs being used for at the radar station? PCBs were used extensively in urban areas with transformers, with electric transformers. PCBs are basically a very thick oil, industrial grade oil, that was used as either in hydraulic fluids or as a transformer oil. And the reason they were such a beautiful product from a commercial perspective is that they could transform electricity or lubricate a heavy machine without breaking down, without warming up, without catching fire. And if you think about the history of the electricity grid, when electricity started to become fashionable in the early part of the 20th century, a lot of our structures were made out of wood. So there were tremendous fires when electricity started to ramp up in our urban areas. So PCBs were a way to mitigate, if you will, the risk associated with fire or of fire. And in the same thing with the radar stations. You have heavy machinery requiring a lot of electricity. There's a risk of fire. You want an oil that's not going to break down, that's not going to catch fire. PCBs were great. Of note today is that mineral oils are used extensively for as a transformer oil or as a lubricant. So mineral oils are often used by the electricity companies to go from the very high voltage to the household voltage safely with minimal risk of fire. So.
Okay, so I'm going to read a question here from Paula through our YouTube channel. Have you studied the seabirds that feed or breed in the same area of the seals? Have you noticed something in the eggshell?
So the question has to do with seabirds. The pigeon guillemots were the only species of seabird that were studied there. And of course, seabirds, if you think about aquatic birds or seabirds or colonial water birds, often these are ones that eat fish or invertebrates. They're connected to that aquatic food web. They're not species that are looking for grains or hanging around croplands, et cetera. They're getting down into the water column. They're diving down. They're swimming around. They're picking up fish. And in this case, the pigeon guillemots did pick up PCBs from that contaminated food web, and that did affect their health. Some of the results that I didn't point to here. In terms of eggshell, I would suspect that the person asking this question is wondering about eggshell thinning. Eggshell thinning was not related to PCBs in the past, but it was related to DDT contamination. DDT, the organochlorine pesticide, also banned about the same era as the PCBs, a year or two later. DDT was an organochlorine pesticide. It was a persistent organic pollutant, so it is one of the Stockholm Convention POPs or POPs, and that was a chemical that very clearly resembled estrogen. So it disrupted estrogen dynamics in female seabirds, and we saw eggshell thinning because of that disruption. And when the females were roosting, they would actually crush their chicks and their embryos before they hatched. When the eggshell got to be, I think it was around 14% thinner than normal. So DDT caused a disruption in calcium dynamics because of a disruption in estrogen physiology, led to a thinning of the eggshell, and we saw widespread extirpation, i.e. localized extinction of a lot of seabirds, double-crested cormorants in the Great Lakes, bald eagles, peregrine falcons, pelicans, a lot of different species of birds that were relying heavily on aquatic food webs were getting exposed and adversely affected by taking in too much DDT across the continent of North America and Europe and Asia. So eggshell thinning, another interesting story, probably the preeminent story of wildlife toxicology if you're looking for population level consequences of impact associated with a single chemical. It's DDT, and it was a nasty story.
All right, and our next question is from Zach, also through our YouTube channel. How many PCBs does it take to contaminate wildlife?
How many PCBs does it take to contaminate wildlife? Well, the interesting thing, that's a very difficult question to answer, but it doesn't take a lot. If we look at Saginaw Bay, for example, it's 450 kilograms of a very, very thick oil, most of which stayed very close to the contaminated site. You saw that red zone where that thick oil stayed, and it took quite a long time for that thick oil to sort of weather and move out away from that source. So PCBs tend not to move very far, but it doesn't take a lot. You saw that single spill led to contamination of the entire food web within that fjord, well beyond the boundaries of the contaminated sediments. It does not take a lot. Yeah, I think I would leave it there.
All right, great. We do have a third question. The question is about, can you talk a bit about the cetaceans? And I'm gonna make the assumption that maybe in the context of contaminants, because we've been talking a fair bit about those. Next, we have some other classes where we'll talk a fair bit about the natural history of cetaceans. So, do you want to make any comments on cetacean contaminants?
Sure. I mean, cetaceans are marine mammals, but within all of the marine mammals that we know from all over the world, there are a number of different life histories. We've got very long-lived species. Bowhead whales we now think could live to 200 years or more. Long, long time. We've got the ring seals that live maybe 35 or 40 years. We've got the killer whales off our coast here, where females can live to be 80 or 90 years. A long, long time. So some of these species were, and some of the individuals within these populations were born before PCBs or DDT were invented commercially. So we've seen they have borne witness to a lot of big changes. If we look at the whales, we've got different types of whales. So when we're looking at contaminants, just think back to that food web. What does the food web look like? Are we talking a gray whale which dives to the sediments and takes a mouthful of mud and pushes its tongue out and filters out all of the different invertebrates and swallows and retains those? Are we talking about humpback whales that might bubble feed and capture herring, a school of herring, and feed on those fish, those smallish fish in the water column? Are we thinking about killer whales, transient killer whales, or Bigg's killer whales, as the transient killer whales here are called, that exclusively rely on marine mammals? So if we have similar ecotypes in the Northwest Atlantic, and we do, and they're feeding on marine mammals, then they're one step higher in the food chain than the ring seal that is already contaminated. So when we look at the persistent organic pollutants, or the PBT chemicals, those with persistence, those with bioaccumulative tendencies, and those that are toxic, we worry most about long-lived creatures at the top of the food chain. That's where we see the highest degree of contamination, and that's why we worry about some of these endangered whales, certainly the killer whales around here, and some other species that we'll hear about in another course.
All right, great, thank you. Nathan had a follow-up question, but Nathan, I'm going to ask for your patience because we're going to talk a bit more about cetaceans in some other classes, so I want you to tune in for those and bring your questions back for those. And a final question for Dr. Peter Ross tonight. Is the melting of the ice affecting the sedimentary process of layering on top of the PCBs? You talked a bit about that layering.
That's an excellent question, and of course the Arctic is changing as we speak, and it's changing quickly. We do worry about what that means, both in terms of the biology and ecology. A lot of these ice-associated species, whether it's ring seals or caribou that have to transit from island to island, or polar bears that are looking to feed off ring seals. So climate change is very real in the Arctic and presents a very grave danger to a way of life of many different species, as well as the Inuit that rely on these. In terms of pollution, though, climate change has the potential to greatly alter the way in which different types of pollutants move around the environment. If you think about pollutants that are traveling through our air, so air pollution, they may be dropped out onto a body of snow or ice, and then that's where they lie until springtime, and then they might evaporate, they might get into the water column. If that ice disappears, those pollutants are going to enter the water more quickly, and other pollutants might evaporate in a different manner. So climate change is going to change a lot of the functions related to transport and fate of different types of contaminants. And that's something we are watching for in the Arctic, no question. In terms of sedimentation and some of the other processes, absolutely. Climate change is going to be changing hydrology, it's going to be changing air patterns, weather systems, it's going to be changing the nature of the permafrost, river runoff, organic material, that provides silt that will then become sediments, and biological productivity in the ocean, which will also create sediments. So any changes to climate are going to impact on all of these cycles that ultimately lead to sedimentation and burial. So we need to learn more, and we have to be paying attention to some of these issues with the best available scientific techniques and methods. But a very good question.
All right, great. Thank you very much, Dr. Peter Ross. How about a round of applause for Dr. Peter Ross?
Thank you.
All right, so we'll wrap up and thank everyone for coming and joining us tonight. The Vancouver Aquarium Marine Science Center is a nonprofit organization committed to conserving aquatic life. If you'd like to support the aquarium and its programs, you can visit our website, www.vanaqua.org. Thank you.